Dear students, the next lecture will be about the development of the respiratory system. But before I start this topic, I'd like to add some data about the COVID infection, about the possible target cells of the COVID virus. These are quite new data within 1 month, and that's why I wanted to add to the histology part of the respiratory system. These are unfortunately quite hot topic now. These are published, or these have been published in two very important and quite good papers. The Cell, which has a higher impact factor than Nature and Science, and the other is in the Neuron. That's why I wanted to add this. Both are within 1 month. The target or the most important factors for the virus infection, it seems the angiotensin converting enzyme 2. This is the receptor of the SARS-CoV-2, and you will study this enzyme in the normal physiological functions for regulating the blood pressure. We will study this in the kidney session, so it's quite early now, but I wanted to add this to the respiratory system. This enzyme normally, the type 1, converts angiotensin 1 into angiotensin 2, which will be important factor for increasing the blood pressure. And this enzyme, the type 2, which was discovered only in 2000, is a regulator of the system, so it reduces the blood pressure. It has a very important role normally. And interestingly, which is expressed in various cells, but among these in the lung as well, in the epithelium. Interestingly, this is the receptor of the virus infection. But not only this factor, but also the serine proteases are important for the virus entry, because this serine protease is able to cleave the viral spike S protein, and this is important for the entry. And based on these two factors, some people here tested the expression of these factors, and I labeled and I listed the target cells where these factors expressed at high level. And that's why these are the target cells for the virus. In the proximal airways, we studied the basal cells, which are the resting cells to multiplicate, and also the ciliated columnar cells. The secretory cells, such as the goblet cells, a question I read in some papers where they found some expressions, but then I found another one which doesn't show this expression, so that's why I put a question mark after this. In the distal airways, such as the alveolus, the major cell is the type 2 alveolar epithelium cell, or pneumocyte type 2, which is important for the surfactant production. And we mentioned this in the histopart earlier, and this is otherwise the precursor of the type 1 epithelium cell as well. Now, what is interesting, and this may explain the loss of the smell and taste, is the target in the olfactory epithelium and the taste buds. In the olfactory epithelium, the basal cells again and the supporting cells or sustentacular cells are the target cells, not the receptors. That's why I highlighted and underlined that no neurons were labeled, or no any neuron expresses the previously mentioned factors. But interestingly, it is present in the Bowman's gland as well. In the taste buds, we have expression in the receptor cells, and also in the basal cells and in the keratinocytes around. So what I wanted to tell with this, the taste bud is a clear thing, because if the receptor cells are influenced and the virus attacks this cell, then can be a damage in the taste sensation. But in case of the olfactory epithelium, interestingly, the neurons and these receptor cells are intact. So here, the sustentacular cells or the supporting cells are infected, and somehow the metabolic activity of these cells on the receptor cells is impaired, and that's why probably the olfaction has also an impairment. I took this picture from the publication, and here it shows the pseudostratified kinase columnar epithelium. Here you see that mostly the ciliated cells and also the basal cells are influenced highest and then the middle part of the airways. In the bronchioles, we see also expression in the ciliated columnar cells and partly in the basal cells. And at the level of the alveolus, we see, sign of expression only in the cubital cells, the protruding cells, which are type 2 pneumocytes. The expression level is getting less along the airways, and that's why the infectivity changes also. So that's why the first step for the virus infection is always in the higher airways. This shows the olfactory epithelium with colors. The receptors are in red and orange. The sustentacular cells are this light brown, let's say, and the basal cells are the yellow. And if you look at the gray scale, which shows the expressions, the receptor cells are empty, basically. So this means that here the supporting cells and the basal cells are influenced the most, also the glandular cells. In the respiratory epithelium, otherwise, almost everything is included. In the virus infection as target cell, the ciliated cells, here the goblet cells as well, basal cells, which is not labeled, is these two. These are the so-called brush cells. I mentioned in my lecture that these are mechanoreceptor cells. This is the same with text, and it shows that the olfactory sensory neurons, so the receptor cells, are quite primitive, so-called primary receptor cells, having bipolar shape. The processes on the top of the cell is found along the cell surface. This is able to bind the chemicals, and the central process will form the olfactory nerve through the cribriform plate, and this is which forms synapse in the olfactory bulb above the cribriform plate. And it shows this, that here the nerve cells are not influenced. This little asterisk shows the infection, and the mark is in the pericyte. You studied in the first semester, pericytes are around the endothelium cells, and this is a very important precursor of several connective tissue and muscle cells. It seems that if we have pericyte infection with the virus, then it may modulate the perfusion of the blood and the local blood pressure. This is the corresponding text. And we studied the tongue as well with the different papillae, and it shows that in the circumvallate papillae and in the fungiform papillae, these are the taste buds we studied, it shows more receptor types than we studied. I just mentioned receptors and sustentacular cells. Here we have also type 1, type 2, type 3, and you see that here mostly the nerve cells, the receptor cells are labeled, and the supporting cells around less influenced or not measured. It means it may be also important. And interestingly, the keratinocytes around are also among the target cells. So this shows that virus may target directly the receptor cells. That's why it can cause transient taste desensation, or the sustentacular cells, such as it was in the olfactory epithelium, and that's why it may lead to smell loss. In the background, we have the function of the ACE2, the angiotensin converting enzyme 2. As I mentioned, this is normal function, and that's why we have a nice balance between the two angiotensin types and enzymes. But if the virus is able to target the angiotensin converting enzyme type 2, then the level will be decreased, and the balance is not normal anymore and shifted to the angiotensin 2 and aldosterone axis, and that's why the pressure, the blood pressure increases, and we have several effects which are not useful, beneficial for the organism. So after the target cell infections, we have a signal to the underlying leukocytes, and mostly the neutrophils and the monocyte macrophages will increase in number. Interestingly, T cells will decrease. And what I want to highlight is the T cell, because the tests based on the antibody production, maybe that's why not so useful, because in many cases the B cells do not react properly on the virus infection, and the major is the T cell response. So as far as I know, in the world there is only one kit with one produced by one firm which is able to detect the T cell. Otherwise it is based on the antibody production, which is not proper. So anyway, macrophages and neutrophils will release several cytokines. You study this later in immunology and physiology. This is the so-called cytokine storm, and interleukin 6 and 10 and the tumor necrosis factor alpha are the major, and this leads to the hyperactivation of the immune system. So hyperactivation, inflammatory process induced, and not inhibition of the immune system, and that is the major problem. And what are the consequences of this? We have this normal alveolus, then we have fluid in the alveolus, immediately decreases the surface for the diffusion of the oxygen. And if it's more severe, then we have protein-rich fluid, and the hyaline membrane is made or produced around the surface of the alveolus, and the barrier between the endothelium and the alveolus wall thickens. We have settlement of collagen fibers. This is kind of scar tissue formation, and significantly reduces the efficiency of the gas exchange. This is the problem. In addition, which is not seen here, it leads to blood coagulation, and we have thrombus formation in the vessels. Here you see the normal tissue, quite clear, empty, and if we have hyaline membrane formation with pink on the surface of the alveolus, of course, significantly reduces the surface of the gas exchange. We have thrombus formation in the vessels. We have sign of lymphatic infiltration, and in general sign of inflammation. So you don't need to know this. I just wanted to tell and show some pictures about this. This was published also in summer. Now then I like to tell something about the development of the respiratory system. First of all, the body surface was not enough for the oxygen amount during the evolution, and that's why we have other organs which develop for this purpose inside, which is in contact with a special media rich in oxygen. First it was water, such as the fish, and later on it became air, but we need an inner and humidified gas exchange chamber, and that's why the lungs developed inside. To understand this process, I like to show you the embryo, and this is the cut, what we wanted to show inside. If we cut the embryo here, we can see twice the embryo in cross-section. First in the cranial part, this is what you see. You see the developing brain vesicles, not important, and this is the rest here, distally. But you see that around the neck region, or a little bit distally, we have arches like thickenings on each side. These are the pharyngeal arches or bronchial arches, and the whole is the pharyngeal gut. Now, in each pharyngeal arch or bronchial arch, we have a light blue area. This is a core for skeletal elements and muscles, and each has a nerve and an artery, what we study later, and they will be distinct questions in the embryology topic. Between the arches, we have approximately 5 + 1 rudimentary arch. Between the arches, we have also mesenchyme swelling. So in this direction, it would be from this view, where we see in the midline some mesenchyme swellings anteriorly, which corresponds to the first pharyngeal arch, but not from the first pharyngeal arch. We have two little tubercles. These are the lateral lingual swellings, and between them, behind, we have the tuberculum impar, which means only a single. They will unite to form the body of the tongue, and posteriorly bit or distal bit, we have another mesonomic swelling, and this is called the mesobronchial area, or you will see on the other slide, hypobronchial eminence. This is the other name, and this will split into two. The proximal part is copula, the distal part is furcula, and the copula will join to these three other tubercles, and it forms the root of the tongue. The rest, so the furcula, will contribute to the formation of the laryngeal inlet. Now this is with higher magnification. You see the three anterolocated tubercles, the two lateral ones, and between them the impar, tuberculum impar. They form the body of the tongue. And a little bit more distally, we have the hypobronchial eminence or mesobronchial area, or proximally the copula, distally the furcula. The copula above will form the root of the tongue. The furcula will play a role in the formation of the laryngeal inlet. Now here you see the corresponding nerves, and it will explain some logical considerations. For example, the first three, which form the body of the tongue, correspond to the first pharyngeal arch, the nerve of which is the trigeminal nerve, the third branch of the trigeminal nerve, which is the mandibular nerve. That's why the body of the tongue is innervated by the lingual nerve as a branch of the mandibular. This is the somatosensory innervation. The facial nerve, the nerve of the second pharyngeal arch, doesn't play a role in the formation of body, but somehow the facial nerve is also involved in the innervation of the body of the tongue as the taste sensation, and it will be carried by the lingual nerve at the end. But this branch which plays a role in this is the chorda tympani. I mentioned this in the previous lecture. The copula, which is the proximal part of the mesobronchial area, corresponds to the third pharyngeal arch, the nerve of which is the glossopharyngeal nerve, and that's why it's logical that the root of the tongue, which is formed from this area, is innervated by the glossopharyngeal nerve, both the somatosensory and the viscerosensory parts, so the taste. The rest belongs to the larynx, and that's why, or the area around the larynx, and the corresponding pharyngeal arches from 4th to the 6th, the nerve which is the vagus nerve, and that's why the vagus will innervate the larynx basically completely. So the major cartilages, the thyroid, arytenoid, and cricoid cartilages, are from the pharyngeal arches, not from the mesobronchial area, from the pharyngeal arches, these three. The epiglottis is from the mesobronchial area corresponding to the furcula. Otherwise, the lumen is first filled with endoderm completely, and then we have a recanalization, and the different parts of the laryngeal cavity will develop later. So that's why we have vestibule, ventricle, and subglottic cavity, and we have the true and false vocal folds. The innervation explains lots of things. The muscles of the larynx develop from the 4th to the 6th pharyngeal arches, the innervation of which is the vagus nerve. That's why, if you remember the gross anatomy lecture, I mentioned that both the extrinsic and the intrinsic muscles of the larynx are innervated by the different branches of the vagus nerve, the extrinsic muscles by the superior laryngeal nerve, the intrinsic muscles by the recurrent laryngeal nerve, the final part of which is called inferior laryngeal nerve. Before I go to the ramification of the bronchus tree, I'd like to explain something, one of the most difficult topics of the whole embryology, the serous membrane membrane cavity development. At the beginning, after the folding, here you see the neural tube in brown, and then the gut tube in orange. Both are curved because of the cranial and distally by the caudal folding. The blue, which is the serous membrane, originally in front of the buccopharyngeal membrane and arch, is migrated to the thoracic region, and you see this as a bulge. This is simply the pericardial cavity at this stage. And as you see here, we have another serous membrane cavity, the peritoneal cavity around the yolk sac. And we don't see now at this phase the third serous membrane cavity, the pleural sacs. Instead, we have here two communications between the pericardial cavity and the peritoneal cavity via the pericardio-peritoneal canals, which are located on each side of the gut. Now, when we watch the foregut, we see a little protrusion in the midline. This is called respiratory diverticulum. And it grows out and descends. At the end, it splits into two. These are the lung buds. But if you could see inside, we have a quite elongated communication between the red and the developing orange. So we have a quite elongated communication and gap between these two tubes. But here I wanted to highlight something. This is maybe the most important statement in this lecture regarding the derivatives of the respiratory tube. The epithelium lining, the inner epithelium lining, of course, of the respiratory tube and the related glands are from the endoderm of the foregut, because the gut is from the endoderm regarding the epithelium and also the gut, the glands. So the epithelium lining and the related glands are from the endoderm of the foregut, and the rest of the wall of the tube is derived from the surrounding visceral mesoderm or splanchnopleural. Here you see this in green. So what does it mean? The cartilage, the connective tissue, and the smooth muscle elements, what we studied in the bronchus and tracheal levels or histology. So then the lung bud grows out, starts to ramify, and it protrudes the adjacent visceral mesoderm or splanchnopleural. And as I mentioned to you, first we have a quite elongated communication between the outgrowth, so the respiratory diverticulum, the blue, and the remaining gut tube behind. And this should be closed by these two arrows, which then fuse, and this is called then the esophagotracheal septum. But if this union is failed, then we may have different types of communications between the tubes, between the two tubes, and this is quite a big problem later. If we have a blind-ended tube, this is called atresia. This is the most common malformation. In worst case, we have communication between the two tubes, and this is the worst when the proximal part of the esophagus communicates with the trachea here or here, because the food what the baby swallows immediately goes down directly to the bronchus tree, causing pneumonia. So should be operated as soon as possible. Otherwise, thanks God, the majority of the malformations happens this way. So we have atresia of the esophagus and we have fistula. I highlighted two important embryonic malformations. One is the atresia, the blind-ending tube, and the fistula when we have communication between two tubes. So basically everywhere we see fistula with the exception of this. Quite a typical symptom in pregnancy and gestation is the excess of the amniotic fluid called polyhydramnion or just hydramnion. Of course, not necessary to be this phenomenon in the background, but if the mother has excess of hydramnion, amnion as hydramnion, then you have to watch this more and more precisely to see whether malformations are behind. The reason why we have excess of the amniotic fluid is the blockade of the swallowing of the amniotic fluid, because the baby during the pregnancy circulates the amniotic fluid, and if we have a atresia blind in the tube, then it's impossible. But in case of twins, for example, the mother has also excess of amnion, amnion than normal, so we have other normally present, situations when the amnion fluid is more than normal. Now, back to the development. I like to tell you something here, which was quite favorite question in first year embryo exam, to make a drawing about this, above the amniotic cavity, below the yolk sac. We are behind, after the neurulation process, so neural tube is separated completely from the surface ectoderm in blue. We see the intraamniotic mesoderm, so we are after the gastrulation as well. The notochord in the midline. This is the axial intraamniotic mesoderm, and we see the other parts of the intraamniotic mesoderm, somitomers or somites. This is the paraxial mesoderm. The yellow is the intermediate mesoderm, and then we have the lateral plate. We have parietal and visceral mesoderm. If you look at the endoderm, it is not completed yet as a tube, but very soon will be. This will be the result of the folding. And in this picture, you see the endoderm tube already surrounded here by this light blue. This is the visceral or splanchnic mesoderm around. And the red arrows show the outgrowth of the lung buds from this tube, from the gut, and that's why it shows that when it grows out into this cavity, the light blue is always protruding around. And it shows that that's why the light blue, which is called splanchnopleural or visceral mesoderm, gives the non-epithelial derivatives, such as the connective tissue, smooth muscle, and cartilage, and also the mesothelin lining of the visceral serous membrane. And as I mentioned, the epithelium and the associated glands of the airways will form the endoderm of the foregut, so from the whitish cell layer. The separation of the body cavity is one of the first topics in embryology, so I don't wish you to draw this question in the exam, but you may be involved in this topic if you get this question. That's why I wanted to show you, to help to understand this process, schematic drawings. So this is the gut, the yolk sac still connected to the midgut, and this is where we have the pericardial cavity, the peritoneal cavity in this region connected to the pericardial cavity with two ducts or canals at this stage, and this is with orange the development of the trachea and the lung buds. And as you see, the green would be the surrounding splanchnic mesoderm or visceral mesoderm, and that's why when the lungs grew out from the foregut, protruded the visceral mesoderm around. This is what you see. Originally, we have this common cavity for the heart tube, and these are the developing lungs from the foregut, and you see the orange around the lung will be the visceral mesoderm. This is later the visceral pleura, and something grows towards each other behind the heart tube. We will study later in the development of the heart. This will be a special septum, which is formed by the elevation of the venous end of the heart tube, carrying the common cardinal veins. These are the vessels here. You will study later with the phrenic nerve, and these septae will fuse and completely separate the pericardium anteriorly from the pleural sacs behind. So that's why we have three serous membranes in the thoracic cavity. One is for the heart, this is the pericardial cavity, and two for the lungs as pleural sacs. This shows the ramification, the generations of the Bronx tree. Altogether we have 22 to 24 generations, not finalized by the birth, just 17 approximately present, and we have still continuation of this process up to the third year. And the first division is around the fourth week. Two lung buds appear, and the principal bronchi. Next week, lobar bronchi. 6th week, segmental bronchi. Don't forget, on the third week, we have very important processes: gastrulation, neurulation, circulation. So fourth week, 5th and 6th week, these are the first three generations of the Bronx tree, all together like this. and the bifurcation will be the first division at the level of the fourth thoracic vertebra. It is not important, but I wanted to include some molecular biology in the lecture. It shows what factors control the bifurcation, because we have here a nice interaction between the endoderm tube, yellow, and the surrounding visceral mesoderm. We have factors which stimulate, which induce the proliferation of the endoderm tube. This is the retinoic acid and the TBX4/5. Through FGF10, they are able to act on the endoderm to proliferate. But if something blocks the effect of this, such as the bone morphogenic protein 4 and the sonic hedgehog, you studied these factors in first semester, then at this area, the endoderm doesn't proliferate, and this will be between these two proliferations. So that's why the bronchus tree divides in a dichotomic manner. These are the phases of the maturation of the lung. First, we have a pseudoglandular period, quite similar to the gland appearance, and it is between the 5th to the 16th week. Dichotomous division until the terminal bronchiolus, then bronchiole. Then we have the canalicular period between the 16th to 26th weeks, when the respiratory bronchioles and the alveolar ducts develop, and we have less connective tissue between the developing vessels and the bronchus tree. And the final is the terminal sac period, 26 to 40, when the vessels are adjacent to the epithelium lining of the alveoli. It should be very crucial to have a very tight contact between them, and this develops by the end, and doesn't finish the process. As I told you, this is the alveolar period, which starts just before the birth, but it extends until the year 3, and that's why the number of the alveoli increases from 50 million to 300. This shows the same. Here you see how the vessels are closer, at the same time how the epithelium is getting thinner to be the type 1 pneumocyte for gas exchange. And it shows two pictures. One is the fetal lung. We don't have this specimen by the exam, but it shows the glandular appearance. And this one is the mature lung with open, alveoli. Surfactant was mentioned last time in the histo lecture. The major cell, which is important at least at the level of the alveoli, is type 2 pneumocyte or alveolar epithelium. And the amount and the timing of the surfactant production is very crucial to have a properly mature lung for gas exchange. And this is the risk of the premature baby situation. The type 2 cells appear on the 20th week, so the middle of the gestation, but the production of the surfactant is sufficient and enough only around the 32nd week. So that's why if we have a premature baby, you have to add maybe a special drug to induce the surfactant production. This is stimulated by the steroid, because if we don't have enough surfactant, very serious condition can develop. This is called respiratory distress syndrome. After the birth, we may add some natural or synthetic components as well of the surfactant, which may help to reduce the surface tension, because that is the problem at the end of the expiration. When we don't have enough surfactant, the surface tension is high to stick the alveolus wall together, and we are not able to reopen it anymore. The components of the surfactant will be listed here. I mentioned this in the histo class. I don't want to recap this. What I wanted to add is this, that type 2 cells are able to differentiate it into type 1. Don't forget, this is here the target cell of the COVID virus at the level of the alveoli. And at the end, I want to add a malformation or two malformations. One is the agenesis. Of course, no way to survive in this case if you don't have lung. The second is a problem after the birth. if we have hypoplasia of the lung. Here I have to say something, a general rule. Two factors are important for the development of the lung, the proper development of the lung. One, the enough space around where the lung is able to grow in. The other is the proper pressure inside. So that's why we have fluid inside in the respiratory, in the airways, to have enough pressure. These two conditions would be the most crucial for the proper development of the lung, so for the generation of the bronchus tree divisions. If somehow we don't have enough space for the development of the lung, in case of the herniation of the diaphragm, when very often the problem is associated with shortened esophagus, and the esophagus appears in the left side of the thoracic cavity, doesn't let the lung properly develop because not enough space in this case. This is called hypoplasia. So it should be operated, and it's relatively frequent in the newborns. Thank you very much for your attention, and we will continue later with gross anatomy.